Hello, I'm Junkai Ho from ATR Computational Neuroscience Laboratories. I'm going to introduce my work inter-individual deep image reconstructions. Human brains are hierarchically organized. When we perceive a visual image, our brain first processes simple features such as edge and orientations in early visual cortex and compose features in extrastriate cortex, eventually leading to shape attribute and even objects identities in the higher ventral visual area. This is similar to a deep neural network which processes an image hierarchically. The brain activity thus contains hierarchical and fine-grained visual information that allow us to reconstruct the visual image via some reconstructions algorithms. However, brain shows individual difference in both microscopic anatomy and the fine-grained organizations of feature representations. The question is, could the hierarchical and fine-grained representations be converted across individuals while preserving encoded perceptual contents? We use a method of functional alignments called neural code converter, which convert the brain activity of a sub-subject into a target subject brain space. The converter is trained with the fMRI response to a sequence of images from both subjects. Especially, the converter is trained with the brain activity of the whole ventral visual cortex. No individual brain area information was assumed during training. We evaluated the converter by computing the Pearson's correlations between the target subject's measured fMRI pattern to a test image with the converted fMRI pattern from the source subjects. The converter achieved mean convergence accuracy at around 0.5. This indicates that considerable amount of information was preserved. Next, we examine if there is a hierarchical correspondence between two subjects' brain area. Recall that the converter is trained with the brain activities of the whole ventral visual cortex. Next, we perform sensitivity analysis on test data set by excluding a source brain areas to check how much performance drops for each target area. The x-axis here is the excluded source area, and the y-axis is the performance drop caused by the source area exclusions. The largest performance drop of each target area was often caused by the exclusion of the corresponding source area. For example, the target V1 corresponds to source V1. Target V2 corresponds to source V2. There is a trend shifting from V1 to HVC. This result showed that the cortical hierarchy between two subjects was detected automatically. Next, we perform inter-individual deep image reconstructions using feature decoders pre-trained on target subjects' brain data. We would like to examine whether the visual image could be reconstructed with the feature decoders trained with different persons' data via neural code conversions. We took a brain activities from a source subject and converted into the target subject brain space using the converters. And we decode the converted brain activities into the DNA features of the test image. And finally, we reconstruct the visual image using the deep image reconstructions. Here we show the reconstructed image. The top right here is the within individual reconstruction, in which the feature decoder is trained and tested with the source subjects on data. The bottom row show the across-individual reconstruction in which the feature decoder is trained with the target subject data, but is tested with the anatomical aligned or functionally converted brain activities from the source subjects. The video showed the iterative reconstruction process. The image reconstructed from the converted brain activities show recognizable shape similar to the objects in the presented images, and show similar visual quality to the within individual reconstruction, and outperform across individual reconstructions with anatomical alignments. These results indicate that the encoded perceptual contents were preserved. Next, we examine how much data is sufficient to train the converter. The feature decoder is trained with all available fMRI data from the target subjects, together with the images viewed in the experiment. Instead, the converter is trained with a subset of all available fMRI data from both subjects. The amount of training data is converted into fMRI data collection time for simplicity. With less amount of training data, the converter could still produce discernible reconstructed images. We showed that the converter automatically detected the cortical hierarchy. We successfully reconstructed images from the converted brain activities. The results show that the hierarchical and fine-grained visual representations that preserve encoded perceptual contents could be converted across individuals. Even with a small amount of training data, the converters preserve minimally sufficient information for visual image reconstructions. That's all for my presentations. Thank you.